Our research has found a new mechanisms for a regulation of the intestinal immune barrier. Mincle is a receptor that is expressed on dendritic cells in the gut and detects bacteria from our commensal microbiota. What we have found is that upon this detection, Mincle induces the Th17 response and lymphoid cells that in turn produce interleukin 17 and interleukin 22 that regulates immunoglobulin A and antimicrobial peptides in the gut that keep our commensals under control.
The intestinal barrier is a structure that limits the location of the commensal bacteria inside the gut. These bacteria exert important function for our health, for example, providing us with nutrients, but when the intestinal barrier is disrupted, for example, by several factors such as the stress, these bacteria escape from the gut, disseminating in our body and causing inflammation, which can drive several diseases, such as, for example, the metabolic syndrome.
Regarding clinical applications, it's important to consider that recent research has shown that many diseases such as obesity, fat liver, metabolic syndrome, or autoimmune and autoinflammatory diseases such as atherosclerosis or lupus are related to systemic dissemination of microbiota that leads to low-grade inflammation. So our work has shown a new target, a new receptor, a new mechanism pathway, and how we can act on immune cells to reduce this dissemination of microbiota, to keep microbiota under control, and therefore this could be applied at the therapeutic or preventive level in these diseases.
So that could be the case, since our study reveals that the signaling triggered through the activation of the receptor reinforces the intestinal barrier. That would be desirable for those stress situations or pathologies which has been demonstrated that there is an increased intestinal permeability. Our study also helps to understand the important role of the immune system in the intestinal barrier, and also confirms the important role of the immune system in many pathologies in humans.